Hello and welcome to Nikolai's genetics lessons, and today we are going to solve a very interesting problem. Here's a problem: two 32 cm plants are crossed, resulting in the progeny of the following ratio: 1 24 cm, 8 26 cm, 28 28 cm, 56 30 cm, 70 32 cm, 56 34 cm, 28 36 cm, 8 38 cm, and 1 40 centimeters, starting with A and going through the alphabet to represent different alleles, what are the most probable genotypes of the parents. And how we are going to solve this problem. Take a look. So we have one plant that is going to be 24 centimeters tall. So one such plant. We also have 8 plants which is 26 centimeters tall. So we have a ratio 1 to 8. We have 28, which is 28 centimeters. So 28. We have 56. 56. Then we have 70 plants, which is 32 centimeters tall. Then we have 56. Then we have 28. And then we have 38 centimeters. And one which is 40 centimeters. So this is going to be our ratio of plants of the different height. So we can also represent these numbers as follows, as graph like this. So here on this side is going to be smallest plants, and here is going to be the biggest plants. But this graph probably going to be misleading. Let me build another graph. So here on this axis we are going to have a frequency, and here we are going to have a size. So this is going to be one extreme phenotype, this is going to be another extreme phenotype, and this is going to be smallest, which is 24 centimeters. We have only one such plant, so our first dot is going to be somewhere here, and our last also would have a lowest frequency of 1, so also the dot is going to be here. And next represent 8, so would have probably dot would be somewhere here, and then 28, so probably somewhere here, then 56 somewhere here, 70, 56, 28, and 8. So here's our graph. It have to be symmetrical, but probably in my case is not very symmetrical, but it have to be symmetrical. Now you see this picture better. So here's a distribution of frequencies of different phenotypes. Again, this is going to be a plant that is going to be 24 centimeters tall, and this is going to be a plant that is going to be 40 centimeters tall. And for example, this plant is going to be 26, 28, 30, 32, 34 centimeters tall, and 36, 38. So now take a look. Two extreme phenotypes are represented in this graph with lowest frequency. Next phenotype is represented with high frequency. So the phenotype that is intermediate between 24 and 40 we would find in highest frequency. And this is common knowledge. Probably if you if your class, for example, 30 people, has 30 people, you would have only one person who is going to be very short, only one who is going to be tallest in your class, but majority of the classmates would be of the intermediate phenotype. It is true for if we talk about animals or humans or plants. Distribution usually looks like this. Distribution of traits. So how we can find under how many genes this trait under control? And in order to explain this, let me show you the simplest representation of what going to happen if we have only one gene. For example, this gene can be gene A, and in diploid organism we can find this gene in two variants, for example, dominant and recessive. So dominant allele and recessive allele A. And in a gene pool we may have following genotypes. For example, capital A, capital A, capital A, small a, capital A, small a, and small a, small a. Normally, you can say that this variance would produce two phenotypes, but here we have additive trait. For example, dominant allele, let's say, would add 5 centimeters to the plant height, and recessive allele would add 2 centimeters. So let's take a look what we are going to get here. 5 and 5, 5 and 2, 5 and 2, 2 and 2. Now you see instead of 2 phenotypes we have here 3 phenotypes. So this is going to be one phenotype, plant that is going to be 10 centimeters tall. Here is second plant that is going to be 7 centimeters tall, and here is going to be third phenotype, plant that is going to be 4 centimeters tall. So instead of 2 phenotypes we have here 3 phenotypes, and you also can say that with additive traits number of genotypes equal to number of phenotypes. I'm saying different genotypes. So here we have only 3 different genotypes and 3 phenotypes. So let now put some numbers here which would be useful for us. So we have one gene, 2 alleles, and we have Punnett square, which is 2 by 2, and which gave us 4 cells, and we have 3 classes. 3 classes of phenotypes. So far we cannot say anything about the pattern. We have also now to try a model with 2 genes, because this model doesn't explain what we see in our problem. By the way, let's count how many classes we have here. We have 1, 2, 3, 4, 5, 6, 7, 8, 9 classes. And with in a model with 1 gene, 2 alleles, we have only 3 classes. So now let's try variant with 2 genes and 4 alleles. So genes can be A and B. Again, gene A can give us 2 alleles, dominant, recessive. Gene B also can give us 2 alleles, dominant and recessive. So let's say one parent is going to be heterozygous for both genes, so going to be capital A small a, capital B small b. We cross with another parent of the same genotype, and such a cross, which we call Dihybrid Cross would give us greatest number of the genotypes in their progeny. And what we can expect here? We can expect, for example, that parent 1 would produce following gametes: dominant allele A and dominant allele B. So capital A, capital B. Another variant would be dominant A and recessive allele B. So dominant A, recessive B. And another variant would be small a, capital B, small a, capital B. And the last variant would be small a and small b. So small a and small b. And because second parent has also the same genotype, that means that we also can build a Punnett square, and genotypes is going to be the same as parent 1. So capital A, capital B, capital A, small b, small a, and capital B, and small a, small b. Now we can build a Punnett square. And in order to save a time, I'm going to stop video here and will fill this table. But before I will do it, let me show you a shortcut. Here is going to be genotype, which is going to be dominant for all 4 alleles. And here is going to be genotype that is going to be recessive for all 4 alleles. Basically, we just need to know this. We can solve this problem with this shortcut. I will explain you later. You would understand this logic. But now, let me fill the full table. Now take a look at this table. So we have one extreme phenotype here. And here is going to be another extreme phenotype. So this is going to be extreme large. This is going to be extreme small. And they are going to happen only once in this table, such phenotypes. All the rest between them is going to be intermediate phenotypes. For example, let me choose another color. Here is going to be another phenotype where 3 alleles are dominant and 1 recessive. And it is not important which 3 alleles and in which order. Here's another such phenotype. Here's another such phenotype. Genotype here equals to phenotype. And now we have another variant where 2 alleles are dominant and 2 recessive. And here's another variant and another variant here. And another phenotype would be phenotype where 1 allele is dominant and 3 recessive. Take a look now. We have one extreme phenotype. Here's another extreme phenotype, extreme large, extreme small. And here's another class. So let's count how many classes we have. 1, 2, 3, 4, 5 classes. We have 5 classes, and as you see, each class represented with different frequencies. So now let's return to our table. So when we have 2 genes, we have 4 alleles. Our Punnett square is going to be 4 by 4 and would give us 16 cells. And we are going to have 5 different phenotypic classes. So now we can compare with a model with one gene. Again, take a look. Here's a model 1 gene, 2 alleles. Here's a model 2 genes, 4 alleles. And now we can see certain pattern. But let's do the same for the model with 3 genes. I'm not going to make another Punnett square. It's going to be too much effort, but we are going to use different logic. So take a look what logic would be. So if we would have 3 genes, our genes can be gene A, gene B, and gene C. So gene A can be of the 2 variants, gene B also can be of the 2 variants, which we call alleles. Gene C also can be of the 2 variants. So now we have a model with 3 genes. So 3 genes would give us 6 alleles. Our Punnett square is going to be 8 by 8 and would give us 64 cells. But how many phenotypes we are going to have? Take a look what the logic is going to be. So with 6 alleles, genotypes possible would be as follows. For example, genotype of one parent can be capital A, small a, capital B, small b, and capital C, small c. When we cross with another parent of the same genotype, we can get a range of different genotypes in a progeny. For example, one extreme genotype would be that progeny is going to be dominant for all 3 genes and would have following genotype. And another extreme genotype would be when a progeny would inherit from each parent recessive allele and would be recessive for all 6 alleles. And we also would have some phenotypes that is going to be intermediate, but how many we can find without building a Punnett square. Just let's use different logic. So we have 6 alleles, and one possible extreme genotype would be when all 6 alleles would be dominant. And we also may have 5 dominant alleles and 1 recessive allele. We also may have 4 dominant alleles and 2 recessive alleles. We also may have 3 dominant alleles and 3 recessive alleles, 2 dominant alleles and 4 recessive alleles, 1 dominant allele and 5 recessive alleles, and we also may have 6 recessive alleles. So now each this variant, which would happen with different frequency, would give us number of possible classes of phenotypes. So 1, 2, 3, 4, 5, 6, and 7. So now we can say that we are going to have 7 classes with 3 genes, 6 alleles. Our Punnett square is going to be 8 by 8, 64 cells. And we are going to have 7 classes of phenotypes. And another important information would be that, for example, with 1 gene, 2 alleles, our each extreme phenotype, so this is one extreme, this is another, we see with a frequency of 1/4. 1/4 this extreme, 1/4 this extreme. 2 by 2, 4 cells, and each with a frequency of 1/4. Now take a look. With a model where we have 2 genes, 4 alleles, our Punnett square is going to be 4 by 4, 16 cells total, 5 phenotypes, and each extreme phenotype would happen only once. So 116 would be frequency. 116 would be frequency of the biggest plant, and smallest also would happen with a frequency of 116. And how do you think what is going to be frequency of the phenotype which is going to be, for example, biggest in if we would have 3 genes, 6 alleles? So the frequency of the extreme phenotypes would be 1 out of 64 and 1 out of 64 here. And for example, what is going to be a frequency of this phenotype or this phenotype? For example, 5 dominant alleles and 1 recessive. And the frequency is going to be, take a look, how many variants we would have. I only would do for this number 2 and 6. The rest would be too time consuming. I just want you to understand the logic to the deepest extent. So 5 dominant alleles would be, for example, capital A, capital A, capital B, and capital C, small c. Another variant would be capital A, capital A, capital B, and small c, capital C. Another variant would be capital A, capital B, small b, and capital C, capital C. Capital A, small b, capital B, capital C, capital C. Capital A, small a, capital B, capital C, capital C. And the last variant, small a, capital B, capital C, capital C. So with one allele that is going to be recessive and 5 dominant, total number of alleles 6, we also would have 6 variants. 1, 2, 3, 4, 5, 6 variants. So frequency of this genotype would be 6 out of 64. And frequency of this genotype also would be 6 out of 64. We have here 1 dominant and 5 recessive. So basically we would have, you just have to replace dominant with recessive and recessive with dominant. Logic is the same. But here we would have much greater number of variants, so we would better not to lose time. Now you know the logic how these frequencies are calculated, and if we have 7 different phenotypes, it doesn't mean that all these phenotypes happens with the same frequency. So we went through models which is 1 gene, 2 gene, and 3 genes. And now we have a certain pattern. Take a look. For example, 1 gene, 2 alleles, Punnett square 2 by 2, 4 cells. Each extreme phenotype is 1/4, 3 phenotypes. 2 genes, 4 alleles, Punnett square is 4 by 4, 16 cells. Each extreme phenotype is 1/16. We have 5 different classes of phenotypes. And with 3 genes, we have 6 alleles. Our Punnett square is 8 by 8. Each 64 cells. Each extreme phenotype is represented with a frequency of 1/64. And we have 7 phenotypes. Now, using the same logic, we can predict what we are going to get with 4 genes. So right away we can say 4 genes would give us 8 alleles, And our Punnett square is going to be 16 by 16. So going to be 256 cells. And we are going to get 9 different phenotypes. Again, if we compare this table, we would see phenotypes would grow as 3, 5, and 7. And we can say that the next variant would be 9. So just plus 2, plus 2, plus 2. But for example, if we compare number of cells, we just have to multiply by 2. So Punnett square is 8, 16. So with 5 genes model, we can predict that instead of 16, we would have 32. 32 multiplied by 32. And number of phenotypes we also can predict using this pattern is going to be 11. So let's check our problem again. And we have here 1, 2, 3, 4, 5, 6, 7, 8, 9 classes of different phenotypes, and we have found that we can get 9 classes if we have 4 genes, 8 alleles. So we almost solved this problem. Now let's only check what the question is. Starting with A and going through alphabet to represent different alleles, what are the most probable genotypes of the parents? So, for example, take a look. Genotype of this plant would be, this is representative of this represent plant with phenotype that is going to be smallest. So with 4 genes and 8 alleles, the genotype of this plant would be small a, small b, and small c, and small d, small d. For example, genotype of this plant, which also would be represented with lowest frequency, but this is going to be the biggest, the tallest plant. So genotype would be capital A, capital A, capital B, capital B, capital C, and capital D, capital D. Now, we know that, for example, smallest plant is 24 centimeters tall, and we have only one such plant. If we combine all these numbers, believe me, we are going to get 256. So this is 1 out of 256. This is 1 out of 256. So it's ideally shows what we already predicted, that with 4 genes, 8 alleles, our extreme phenotypes would be 1 out of 256. But how do we know how each allele would add to the trait? And take a look. For example, we know that plant that is 24 centimeters tall has this genotype. And this genotype has 8 alleles. So what we can do, we can divide 24, this is 24 centimeters, by 8 alleles. And we are going to find that each recessive allele adds 3 centimeters to the plant trait, which is plant tallness. Now, let's check, for example, genotype, which is also happens once here, and it is 40 centimeters tall. And we know that genotype would be as follows: 8 dominant alleles. So how much each dominant allele adds to the trait? 40 centimeters divided by 8 alleles would give us 5 centimeters each dominant allele add to the trait. Take a look. For example, we can add 5 here, 5 here, 5 here, 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 and here. We are going to get 40 centimeters. And here we add 3, 3, 3, 3, 3, 3, 3, and 3. And when we add all these numbers, we are going to get 24 centimeters. And let's return to our problem again. We are told that two plants, 32 centimeters tall, were crossed. So what is the genotype of the parents? Just let's guess. If we have the whole range of the genotypes in the progeny, parents have to be heterozygous for each gene. So genotype of the parents have to be capital A, capital B, small b, capital C, small c, capital D, small d, and the second parent also have to be of the same genotype. Capital A, capital B, small b, capital C, small c, and capital D, small d. Let's check. We know that each dominant allele add 5 centimeters to the trait. And we know that each recessive allele add 3 centimeters to the trait. So how tall is this parent? So 8 plus 8 plus 8 plus 8 is going to be 32 centimeters. We are told that two parents who are 32 centimeters tall were crossed. And we got the whole range of different phenotypes with different frequencies, and basically we have solved this problem. What we see here exactly describes the problem, describes the frequencies, describes genotypes of the parents that were crossed. We have found how many genes control this trait, and basically I hope now you would understand this type of problems, and this is type of problems which we call problems with additive trades. Basically, when you do plant breeding or animal breeding, most of the traits would be additive, not like probably what you taught in textbooks. For example, when we have one dominant and recessive allele, which would give us two phenotypes, dominant and recessive. Most of the traits, for example, such as tallness or weight, or for example, if we talk about people, skin color, all these traits would be additive traits. But of course, some traits can be just dominant and recessive, and there are also many other conceptual ideas how genes works. For example, it can be that one gene would be also influence expression of the other gene, not through the dominance but through the epistasis. And there are many different variants also exist, but I hope now you would understand this type of problems much better. By the way, there is a formula, and you can solve this problem under 2 minutes, maybe under 1 minute, but I on purpose gave you this full lecture on this topic, so you would have a deep understanding, and you know now how to understand the patterns, numbers, and how to do shortcuts in order to solve problems. Not necessary each time to build a Punnett square, just follow the pattern. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day, and see you in the next video. Goodbye.